Here's live. This is Mike. Can everyone hear me? All right. Bear with me because I started losing my voice last night and it's just barely there. So if I start squeaking, let me know. All right. Thanks, everybody, for joining us this afternoon. I know everyone's sleepy. You have a full stomach, so I'll try to take it easy on you. I'm just going to say up front, it's very difficult to put together a 10 to 12-minute talk about a 10-year-old company. There's a lot I'd like to share, so if anyone wants to speak afterwards, I'm more than happy to do so. So thank you so much for inviting me here today to speak about ArunA Biomedical. Historically, ArunA is a manufacturer, supplier, and contract service provider of human pluripotent stem cell-derived neural cells. Excuse me. We were the first company to commercialize a human pluripotent stem cell product, derivative product, and we did so in 2007. Excuse me. We are commercial licensees of Worth, and we have exclusively licensed a patent from the University of Georgia for our neural stem cell differentiation process. So ArunA is not a new company, as I alluded to, and the magic of our company is that we have been able to grow over the course of the last 10 years and build a product portfolio and service portfolio primarily through contract and product sales revenues and non-dilutive sources of funding. Our operating and manufacturing services unit has rigid quality guidelines and enables us to be key players in the translational research market. And I'm so sorry about this. Our customers are a who's who among academia, industry, and pharma. And I'd like to point out a success that we're quite proud of, and it is that under a contract with Pfizer, this information, part of this research is published also with Pfizer and ArunA. ArunA did a screening of a small library of compounds. One of those compounds is now in human clinical trials. The quality, utility, reliability, and versatility of our products has enabled us to create a unique sensor element platform that we call cell-based biosensors. You can see on the left side of the screen a sampling of our product portfolio. What we have been able to do is marry our products into proprietary assay platforms that we call cell-based biosensors. ArunA's products are arguably the most widely published human pluripotent stem cell-derived derivative commercial product. We have more than 60 application areas that our cells have been used in, and over 130 peer-reviewed publications from our customers and ourselves. So that's a really quick snapshot of ArunA Biomedical, our reagent platform, but what we're doing now is expanding our 10 years of manufacturing experience in this neural area to include therapies. The first therapeutic area we'll concentrate in is stroke. Excuse me. So I will be focusing the remainder of my presentation, sharing our plans to move into the neurotherapeutics area in the area of stroke. We'll build on our industry-leading position in stem cell manufacturing and build upon a world-class scientific leadership in neural stem cell scientists, and then integrate that into industry-leading preclinical animal models of human disease. We are especially excited about the exosomes that our human neural progenitors produce. We're calling, and we have branded them, NPECs. So ArunA's NPEX exosomes are the result of a natural process They are secreted by the HNP1 cells and transmit signals to other cells, including neighboring cells and distant cells. And the beauty of exosomes is that they have the capability of crossing the blood-brain barrier. We anticipate that the exosomes will be our initial therapeutic product because they have the potential for being superior or as effective as neural progenitor cells at repairing damaged brains. And importantly, they are easier to produce, store, and maintain. So not all exosomes, stem cell exosomes are the same. While it's not fully appreciated that the source and level of differentiation of stem cells matters, ArunA's NPECs are unlike any others because of our proprietary patented position. For example, there are ongoing clinical trials using stem cells in an attempt to restore lost brain function in stroke. For most of these trials, the stem cells come from bone marrow that are called mesenchymal stem cells. One initial experiment that we've been conducting is to compare the exosomes from ArunA's human neural progenitor cells to exosomes from bone marrow, not dissimilar to what others are developing. What we found is that the NPECs exosomes were far superior in vitro, in an in vitro model in a stressed assay. So we're focusing on a major medical issue called stroke. Look around the room, there's probably a lot of us who have been affected by stroke one way or another. So let me explain to you the magnitude of stroke in the United States alone. Stroke every year affects 800,000 Americans. Worldwide, probably multiply that by 2. Of them, 120,000 individuals die. 280,000, thankfully, are spared permanent disability, but half are left with a debilitating long-term, with debilitating effects needing long-term care. The impact on our healthcare system is right in the neighborhood of $30 billion annually. Assuming a per-person cost for treatment of $5,000 to $10,000, we believe that this is a $2 to $4 billion per year addressable market. So let me tell you a little bit further about some of the key studies validating the use of ArunA's platform technology for stroke treatment. We have reason to be optimistic about the ability of ArunA products to repair damaged brains. In a third-party study that was published, ArunA's HMP1 cells were used in a rat model for stroke. The middle cerebral artery was permanently occluded, leading to a massive stroke. 3 weeks later, ArunA's HMP1 cells were directly injected into the damaged area of the rat brain. When looking at the brain grossly in these images and in the stained cross-section of the brain below it, you'll see on the left of the control that the area has been repaired in the HNP1 treated side of the brain. Sorry about that. Importantly, in this study, the repair led to improved sensory motor function through balance and movement tests and cognitive function as well, using a T-maze. So given the in vitro data and this in vivo data, we expect NPECs to be superior to mesenchymal stem cell exosomes since they were superior in studies in tissue culture. While rodents are helpful as an identifying screening tool for treatments for stroke, their brains aren't fully predictive of human response. And leading experts in the field have identified that the rat model is appropriate but not sufficient for therapeutic development for stroke, and has recommended that the pig be utilized as a preclinical model as well. And the most comprehensive pig model in stroke was developed at the University of Georgia by Franklin West, ArunA co-founder Stephen Stice, Dr. Simon Platt, and others. We are fortunate to have this model available through these collaborators at UGA in order to expand our preclinical studies to include the pig. So why the pig, you may ask? Well, in the pig, size matters. The pig brain, while smaller in size, consists of this very similar gray and white matter. In the pig, white matter is approximately 60%, 60 to 65%, and the same as in humans, while a rat is less than 10%. In a stroke model of the pig shows similar to humans, post-stroke and edema in the brain in both gray and white matter. The pig's brain can be easily assessed visually through MRI to monitor progress. And utilizing a specialized pig track and software, the group is able to assess changes in motor function like swing time and stride length. These studies will also assess changes in behavior, learning, and memory in open field tests and T-maze. So we know there's competition, and we know the race is on, and ArunA is aggressively and efficiently moving our therapeutic technologies forward. Of those companies currently pursuing this area, we know that no clinical trials have been initiated using exosomes to treat ischemic stroke. ArunA has a highly experienced executive team. I have more than 15 years of biotech operations experience. I've also spent simultaneously 15 years of public relations and marketing experience in biotechnology. Our founder, Dr. Stephen Stice, has over 20 years of biotech R&D. He's co-founder of 5 biotech companies, 2 of whom are currently in human clinical trials. Mr. Harold Ingalls has 40 years financial leadership. He's a former CFO of Serologicals Corporation, former CFO of La Roche, and former vice president CFO of CardioMEMS. All right, so our strategy, our goal is to develop NPEC stem cell-derived therapeutics as best in class for the treatment of serious neurological diseases, beginning with stroke. Our strategy is to validate and enhance the probability of clinical success by integrating early into therapeutic discovery program. We expect to realize value for the technologies through mergers, acquisitions, or out-licensing. All right, so we're doing this rapidly. We're rapidly moving into these animal model studies. We're beginning in January. We're doing an initial bridge financing right now of $2 million. We're about 1/3 of the way there, and we'll follow that by IND following and a Series A financing. So we're healing a broken brain. I appreciate your time today and would be more than happy to answer any of your questions.
Thanks, Tracy. We're going down the line. Next we have Lee Buckler from RepliCel.
Somewhere there's a cursor. No. There we are. All right, thank you for being here, and to the organizers for the opportunities. I'm here going to speak to you about RepliCel Life Sciences. It's a publicly traded biotech company based in Canada, traded on the Venture Exchange and the Toronto Stock Exchange and the OTC. I will make some forward-looking statements today. So we are a regenerative medicine company using cells as the basis of our therapeutics. The goal of our programs are to address specific deficits of cells or their functions for various clinical indications. We're in the clinic now for chronically injured tendon, We're looking to restore damaged skin and rejuvenate hair growth. We have two distinct cell populations that we have into the clinic. And also, in addition to the biologics, we have a next-generation dermal injector device that we're very excited about in the near term. Philosophically, we are a company dedicated to building high value in mid-stage clinical assets and then partnering those with companies of much bigger size and with commercial prowess. So we don't aspire to take products to the market. Proof of principle there is the one partnership we have already in play with our most mature product with a company called Shiseido, company based in Japan, which is the third or fourth largest cosmetic company in the world, which is co-developing a product for us and with us exclusively in Asia. So we have these three clinical programs. We're in phase 1/2 in chronic Achilles tendinopathy, phase 1 in Germany for aging and sun-damaged skin. We've completed a phase 1 in androgenic alopecia, and we're about to move into next phase clinical trial of that product with Shiseido in Japan, which, as many of you will appreciate, is a very exciting jurisdiction to be doing clinical trials. This is a simple schematic of our therapeutic and manufacturing programs. We are, as I said, an autologous cell therapy company. We derive both of our cell populations from a single suture biopsy from the back of the patient's head. That tissue biopsy is sent to the manufacturing facility where we mine one of two different cell populations. The cell population at the base of the hair follicle that you see there is the dermal sheath cup cell population. We believe ultimately responsible for the cellular milieu that grows hair. And it's why, and it's that cell population that's decimated by those people who suffer from androgenic alopecia. And as that cell population disappears, the hair fiber diminishes. It's the same cell population that when you transplant it along with the whole follicular unit from the back of the head, because you will have observed that most people who have pattern baldness still have hair at the back of their head, and evolutionary reasons which were not entirely clear to us, the cells and the whole follicular unit are immune to the disease at the back of the head. That's why you can take the whole follicular unit from the back, transplant it to the top, and it will continue to grow hair for the rest of your life. We're looking, instead of doing a whole follicular transplant procedure, to mine the cell population that's insensitive to the androgen hormone, grow more of them, transplant them into the top, where we see in animal models they migrate to the base of the hair follicle and regenerate the cellular interactions that grow hair. So that's the one cell population. That is a product-specific technology that we're singly focused on androgenic alopecia for. The other cell technology is a standard, in many ways, fibroblast cell that is responsible for the maintenance of the sheath of the hair follicle. And it's unique in one particular aspect that we're looking to leverage, and that's its high expression of type 1 collagen. Much higher in its type 1 collagen expression than skin-derived fibroblasts, for instance. We have a very simple philosophy of life. We don't differentiate cells. We don't modify cells in any way. We find a cell population. We think we understand what it does. We grow more of them, and we inject them locally, not systemically, to have a local effect. So we grow, it's a simple cell expansion play. We cryopreserve the cells in vials and ship them back for re-injection. And in the case of the scalp and the skin, we have this proprietary cell and dermal injector, which I'll give you a sense of in just a moment. So I'll walk you briefly through the clinical pipeline. The first that we moved into the clinic last year, we had a very exciting year this year, a pivotal year where we moved all of our assets forward to set us up for a really potentially transformative year in 2016 with data, first of all, from this tendon trial, data from the dermatology trial, and a CE mark on our device. The first program that we moved into the clinic this year was for chronic Achilles tendinosis. This is chronic tendon injury, people who are refractory to physiotherapy, refractory to rest, refractory to any other kind of biological injection or ultrasound. They're left with some level of chronic pain and dysfunction. So it's a phase 1/2 clinical trial at the University of British Columbia. Primary endpoint is safety, but efficacy measures, we're measuring several efficacy endpoints, including function, pain, V's and VAS scores, biomechanics, blood flow, not the least of which is ultrasound. So this is actually the ultrasound image of a phase 1 clinical trial done by one of our clinical collaborators using a skin-derived fibroblast. Again, notably inferior in terms of its type 1 collagen production, but nonetheless very akin to the fibroblasts that we're using in our clinical trial. And in 3 small phase 1 clinical trials in the Achilles, patellar, and lateral epicondylitis, he showed very nice restoration of the tendon, as you can see by this image. What you see in the top of that image is the black is is scabbed tendon, chronically disrepaired tendon, highly associated with loss of function and pain. And what you see in the image below is what you want to see in a very healthy tendon. It's long linear strands, well organized, of type 1 collagen, which are indicative of a strong functioning or pain-free tendon. And that's the same tendon from the same man 6 months after a single injection of skin-derived fibroblasts. And to nice statistical significance, restoration of function and elimination of pain in each of those phase 1 studies. So it's that same so that trial is enrolling, treating patients. We expect to read out data late next year, and we're very excited about that data and confident that we'll see a signal because of that predicate work done by David Connell and others in London. It's that same fibroblast cell population and the same reliance on the type 1 collagen expression that we're moving into, or that is in a phase 1 clinical trial in Germany for the potential treatment of aging and sun-damaged skin, on the premise that these fibroblasts will, when injected into an area where the extracellular matrix has thinned and waned, do what they do best, and that is express the kinds of proteins that rebuild tissue. And what you see on the left is the extracellular matrix of someone young, stained for type 1 collagen, and in the middle image you see that, again, extracellular matrix stained for type 1 collagen of someone who is aged. And it's simply because your cells are as tired and old as you are. And so what we've done in this phase 1 study is very interesting, because dermatology is filled, as I am fond of saying, with a lot of pseudoscience and fuzzy endpoints, but we're really interested in biologically measuring what's happening under the skin. So we're doing before and after biopsies. We're using healthy volunteers in a clinical trial in Germany. We're doing 4 injection points, single, double, triple, and injections, along with a sham. Both of these clinical trials are randomized, placebo-controlled. And what we're particularly interested in is the post-injection biopsies against baseline, measuring some of those biopsies materials by histopathology to really assess whether or not we've made a material difference in the extracellular matrix in that around that injection point. And then the other biopsies sent for gene expression analysis against 10 different biomarkers, which are in the published literature highly correlated with aging and sun-damaged skin. And again, that trial is enrolling and treating patients or volunteers, and we expect to read out data late next year. So if we can, in fact, show that we can repair the tendon of your ankle, there's every reason to believe, of course, with some unique, perhaps product tweaks and tweaks of the clinical delivery, the tendons of your knee, both tendons in your elbow, etc. So it's a pipeline product. The product that the company was originally built around is these dermal sheath cup cell populations for the treatment of androgenic alopecia in men and women. It's a disease that often is associated with men, but this is in fact a 42-year-old woman who I spoke with not that long ago, who, as long as she can remember, has had terribly thinned hair in the top of her head. She remembers as she relates the kids driving the cars down the highway in the middle of her head. Because she's always suffered from tremendously thinned hair, and it's genetic in her family. So when we published the phase 1 data from our clinical trial, we saw a pocket of responders that was just as good or better than standard of care, which is either hair transplant or Propecia, which is a repurposed prostate drug contraindicated for women. You get an increase over baseline, but you don't stop the baseline progression. So eventually an increase over hair density doesn't matter anymore. And it's a drug, so it has side effects, not the least of which is erectile dysfunction. Or Rogaine, again, you see an increase over baseline, but not an elimination of the progress of the baseline. So when we published that data, Shiseido company was very interested in discussing potential collaboration with us, and long story short, ended up in a collaboration signed in 2013. It was characterized as a $35 million deal, $4 million up front, $31 billion in commercial milestones, and then sales royalties on top of that. But what's more important to me, in fact, in this relationship, which has been a tremendously productive and collaborative relationship, is that it is also characterized by a commitment by Shiseido to fund all the clinical trials in their market, and their market is Asia, all the manufacturing, the regulatory, the clinical, and sales, marketing, and distribution. So the next phase trial of this product for androgenic alopecia is about to launch in Japan, which it turns out is now perhaps one of the most exciting areas in the world to be commercializing cell therapies. Because, as I'm sure many of you have heard over and over, Japan has made that country potentially the fastest country in the world to bring cell therapy products like we're developing to market. And so there's a potential for this phase clinical trial, for this phase, or this clinical trial that has just launched to be pivotal and to be the trial that launches this product in Japan, and is the reason why, again, akin to our business model, we're pushing very aggressively to co-develop and license our other products, namely the tendinopathy and dermatology products in Japan as well. So lastly, I'll just touch base on the dermal injector. The dermal injector, we became increasingly convinced as we looked at developing our biologics, particularly injecting the cells into the scalp, that we needed to not be susceptible to the variances of clinical delivery. So we invest tens of millions of dollars in developing the best possible biologic product, and if we leave the outcome of that to physicians that you try to train, you leave yourself susceptible to their particular talent or skill. So we wanted to find an injector that you could control the depth and dose and take as much of the art or skill out of the injection as possible, and much to our chagrin, no such injector existed. And so what we've come up with here, this is the second version of the device, which we're very excited about, is the first dermal injector in the world where you can control the depth and dose of delivery. So this is a touch screen. The wand is attached to a touch screen. You name the protocol. It dictates how deep the needles penetrate. It dictates how much those needles deliver per each dose. And you can put absolutely anything in that cartridge. We've designed it to be optimal for our cell suspension, but you can put Restylane, you can put Botox, you can put drugs, you can put vaccines, you can put anything in here and deliver that in a way which is currently not supportable by current injection technologies. And the second really cool value add here of this tool is this funky U-shaped element that detaches from the bottom. This is called a Peltier Element. It's classic cooling physics that's used in all kinds of electronics. No one had thought of using it in conjunction with a dermal injector. So this is where some very unique intellectual property resides with the injector. Essentially, it creates numbing cold on the exterior plate of that element. So when you touch it on the skin, within 2 or 3 seconds, it numbs the skin sufficiently that the needles penetrate, deliver per the depth and dose, and you move and repeat, and you obviate the need for local anesthetics, which most often are associated with dermal injections and have a fair amount of pain associated with them, time, and cost. And so you significantly better the patient experience, you reduce the amount of procedure time, which is important to the doctor, and you facilitate potentially the delivery of things in a controlled manner, which delivers more robust outcomes, but also potentially enables for the delivery of things that currently aren't enabled. If you can do very shallow, for instance, and controlled delivery of product, you are potentially able to address things aesthetically that current injection technologies don't allow. So this is the last slide. 2016 looks very exciting for us. We expect to initiate the pattern baldness trial in Japan with Shiseido. We expect a CE mark on that dermal injector device next year for the use of the device in conjunction with the injection of hyaluronic acid. So that'll be a licensing partnership asset ready for a deal next year. And then clinical data, as I said, from our tendon trial and our skin trial, all of which we think are licensable events and have the potential to bring in significant non-dilutive capital into the company in addition to the sale of equity, which we continues to be ongoing.
Thank you, Lee. Any questions before we move on? Okay, next is Rebecca Marshall from CellectCell.
All right, good afternoon. My name is Rebecca Marshall. I'm the president of CellectCell, and I'm excited to be here with you to spend the next 10 minutes or so introducing you to our new company. CellectCell is an early-stage company focused on developing and commercializing novel tools for cell selection and isolation. So we're a bit different from a number of the other companies that have presented today that are offering exciting therapies for the future. Ours is a tool-based approach specifically for cell selection and isolation. And based on the principles of microfluidics, we've developed a platform that isolates cells with high specificity, sensitivity, and purification efficiency. We're a very new company formed earlier this year and based here locally, based on technology out of the Georgia Institute of Technology. The inventors of the cell selection technology, together with Medtown Ventures, formed this new company, CellectCell, earlier this year. Medtown Ventures is also a local firm. We are a venture and commercialization and development firm that provides management services to new companies, actually to a variety of companies, for things everywhere from turnaround activities all the way to tech transfer, as is the case with CellectCell. So again, we're focused in a slightly different area than many of the other companies that have presented. But as you know, stem cells are the targets of lots of different exciting activities. We've heard of a number of them this week, from basic research to reprogramming as diagnostic tools, and then hopeful therapies for the future. All of these activities start with isolated cells, and that's where we're focused with this technology. The global cell isolation market is large and growing rapidly. As the stem cell market continues to grow and evolve, so will the isolation market. We believe this makes it a prime market for investment and innovation. There are a number of methodologies available today for cell selection and isolation, but they all have some limitations. Manual methods, while they produce high-quality cells as part of the isolated population, are very dependent on the skill and proficiency of the technician and can be subjective and time-consuming. Chemical methods result in dissociation of multiple cell types at one point, giving you, at the same time, giving you limited purity of the isolated cell population. They may also result in cellular changes or even spontaneous levels of differentiation that introduce variability to the research. Most methods do not allow for isolation of intact colonies or serial isolation of multiple cell types. And then many of the newer methods require specific media, reagents, instrumentation, or complicated protocols that can be expensive and time-consuming. Our solution is a disposable cell selection chamber that you see here in this picture. It's based on the natural adhesive signature of cells as well as the principles of microfluidics. And I'm not going to talk at all during this presentation in the interest of time about all the science behind the natural adhesive signature of cells. Dr. McDevitt presented on that earlier today and is sitting in the back of the room if anybody has questions about that specific research. But basically, using these principles, we're able to rapidly and reproducibly isolate specific cell types, again, in a highly sensitive and specific manner. So the way this works is that you would culture your cells under normal standard cell culture conditions. Then you would seed them into the microfluidic channel that runs the length of this chamber. Once they're seeded in the channel, you would incubate the chamber until the cells are adherent. And that's an important point because this device, specifically for research, is designed for adherent cell types. So once the cells are seated in the chamber and are attached, then you apply a fluid flow at a predefined rate depending on the specific cell type that you're trying to isolate. And that's a rate that we would provide to you based on our experiments and the type of cell that you're looking for. And then as the fluid flows past the cells, it's going to actually pick up and collect the cells of interest to be collected at the outlet port for either further cell culture or for analysis. For those of you that aren't familiar with microfluidics, it's the science of manipulating fluids through a small network of channels, and it's recently gotten some attention as a powerful tool for cell culture and isolation because of some of the specific advantages it provides. It doesn't require a large quantity of reagents, it's useful with a small sample volume, and then offers excellent reproducibility while being very gentle on cells. The channel portion of the selection chamber is see-through, which enables you to put it on a microscope and be able to visualize the detachment of the cells that you're collecting throughout the process. So this video actually shows an experiment where the chamber was seeded with fibroblasts and human pluripotent stem cells, and then the flow of the fluid, as you can see, is from the bottom of the screen to the top of the screen, again at a predefined rate specifically for iPSCs. And you can see as the fluid flows through this chamber, an intact colony is actually being gently lifted to be collected in the outlet port. Again, it's based entirely on the natural properties of the cells. This is not enzymatic action. There's nothing else added to this. You can see from the time course at the bottom, the collection procedure is quick and straightforward. It happens for iPSCs in a little under 10 minutes. It's as fast as 5 minutes for some other cell types. And when we look at the isolated population, what we see is, in this case in particular, we got 99% of the cells of interest isolated in this 10-minute collection procedure on the outbound end. Again, this specific device is focused on research scale activities, so the blue circle that you see here is the application segment where we will be initially focused with our first generation product. We do think the platform has applicability to a broader variety of applications, but we're very focused at this point in getting a first generation product out and onto the market to begin generating revenue. We do believe that the CellectCell device offers some specific and unique capabilities compared to other methodologies. Rigorous testing has shown that we're able to isolate the cells of interest with a sensitivity and specificity of more than 95%. Single cells or intact colonies, depending upon the cell type that you're isolating, can be isolated with a very high survival rate of greater than 90%, and those cells aren't being changed as part of this collection process. We've done epigenetic protein expression and gene expression testing. Again, I'm not showing any of that data in this presentation, but it is all available in the reference that you see there at the bottom. But that testing shows that the isolated cells are the same as the initial target cells. And because more than 95% of the cells of interest are collected in about 5 minutes, 5 to 10 minutes for most different cell types, it is a highly efficient process that reduces both time and waste. It's also a very versatile process. The process can be used to either positively select for specific cell types that you're looking for, or negatively select out the things that you don't want, leaving your cells of interest seated in the channel within the cell selection chamber. We've successfully used the technology at this point on many different cell types: induced pluripotent stem cells, endothelial cells, cardiomyocytes, neural cells, and then most recently cancer stem cells. The results of that experiment are actually exhibited on a poster that you can find outside. And when testing the robustness of this technology, we found that we can successfully isolate the cells regardless of the extracellular matrix coating that's used, regardless of the cell line, whether it's human or animal, from both heterogeneous and homogeneous starting populations, and regardless of the cell seeding density. So whether it's a cell that occurs in large quantities or a rare cell, this technology is able to isolate it with good survival and efficiency. We also took a look at the effective passages on the ability to use the technology, and even after multiple passages, what we see is a consistently high purity population of the isolated cells compared to other batch-based methods where you start to see spontaneous levels of differentiation. In talking to potential customers and a couple of focus groups about this technology, they have pretty unanimously responded to two key benefits of this compared to other methods that they currently use, and those are speed and cell survival. The 5 to 10 minute collection process combined with the greater than 90% cell survival are significantly better and easier than methods that they typically use today. Again, it's also, because of the microfluidic principles, capable of working with very small sample volumes. You can serially collect multiple cell types from the same sample. So if you start with a low fluid flow rate and collect a lightly adherent cell and then increase the fluid flow rate to collect a more adherent cell, you can actually isolate populations of multiple cell types from the same initial sample. And then again, as this industry as a whole continues to grow and evolve, there's going to be a continued need for cell isolation because it is a disposable product. As it becomes adopted, we project consistent usage, and that will translate into frequent purchases. Just to give you a little bit of history behind the company, this endeavor all began in 2009 with an NIH grant, through which researchers at Georgia Tech learned that iPSCs, progenitor cells, and differentiated cells all have distinct adhesive signatures. And based only on those adhesive signatures, fluid fluid flow could be used to isolate iPSCs from differentiated progeny, partially reprogrammed cells, as well as parental cells. And all of those things could be accomplished without changing the phenotype or genotype of the cells of interest. That research then led to a couple of patent filings and a publication in Nature Methods. We licensed the technology, gained an exclusive license for it from Georgia Tech in 2014, formed a company earlier this year, and here we are today. Our management team has more than 70 years of experience in life science, medical device, and regenerative medicine, company management, commercialization, sales, operations, and product development. Our 3 founders, likewise, have more than 70 years of experience in stem cell research, engineering, biomanufacturing, and startup of new companies. So we're very excited about the team that we've collected here. We believe the technology addresses a need in the large, rapidly growing cell isolation market. So again, as a tool and a starting point for all of the other stem cell activities that we've learned about this week. We are busy fortifying our IP position. Our experienced team has taken the proof of concept and translated it at this point into a functional prototype that we will begin beta testing in the next few months, with the anticipation of the first product launched in 2016, where we'll begin generating revenue. We're capitalized through launch at this point, but are exploring partnership opportunities both for sales and distribution of our initial research scale device, as well as for future portfolio growth and expansion. I'd like to thank our collaborators and partners. We would not be here today without them: Georgia Tech and the Georgia Tech Research Corporation, the Georgia Research Alliance, and the University of California at San Francisco. I thank you for your time and attention, and I will entertain any questions.
Thank you, Rebecca. Wade is next. Wade Lange from NeuroFx.
All right. Okay, I'm a Mac user, so let's figure out how I cut this out. How do I shut this down? All right. Sorry for, between being a pharmacist and a Mac user, this is always a struggle. There you go. There you go. Okay. Thank you very much. And now how do I go to slides? I'm sorry for my lack of facility. How do I go to slide view? Right there.
I'll get it for you.
Okay, so don't invest because I'm a computer user. So, well, thank you all very much. My name is Wade Lange. I'm the president and CEO of NeuroFx. I'm always intrigued by the kind of commonalities that companies have as we hear. We'll be talking about stroke and a derivation of stem cells. Medtown was involved in the formation of our company many years ago, and I have relatives in Canada. And my best friend's moving to Ottawa. So anyway, so thank you all very much for being here today. Very happy to tell you about NeuroFx. We're a regenerative medicine company and developing a platform technology. So I'm not a scientist, so I won't spend a lot of time on science today, but as many of you know, the science of stem cells appears to be moving away from purely the effects of the cells, but to the effect of the factors that are secreted by the stem cells. We've heard about exosomes. I'm going to talk more about soluble growth factors. My colleague Keith March, who many of you might know, always uses the metaphor of coffee. Most of us probably enjoy coffee, and we like it for the aroma and the flavor, the caffeine jolt, the warmth of the mug. But we don't have to buy raw beans to get that. We can go and buy from our local Starbucks the coffee, and that's essentially what we're trying to deliver here. Is rather than dealing with the cells that we don't necessarily want, we want to capture the benefit of that the stem cells secrete and how they work. So our product is derived from adipose derived stem cells. We essentially, in a manufacturing a GMP setting, grow up the cells, allow them to secrete the therapeutic factors, and we get rid of the cells. So we're left with a liquid that we concentrate down that would be administered via injection to humans. Importantly, as the name would suggest, we're initially focused on neurological uses. The product contains at least these, and we know it, a number of proteins, but BDNF, GDNF, HGF, NGF, and VEGF, all of which are known to have neuroprotective and neurotrophic effects. We believe then by focusing in on the therapeutic factors rather than the cells themselves, we capture the benefits of stem cells, but we reduce some of the potential drawbacks, everything from potential immunogenicity of the cells themselves to a much more cost-effective manufacturing product, so lower cost of goods. An important in acute conditions, an immediate effect rather than waiting for the cells to grow in the body and secrete their factors. The factors are immediately available and again important for acute conditions. And lastly, this is a product then that fits very well into the pharmaceutical business model. They need a bottle that can be on a shelf that is ready to be sold and to be administered to a patient, and that's what we will be able to deliver. Initially a frozen product, but eventually leave a lyophilized powder that will be kept at room temperature in a pharmacy shelf. As a platform technology, there are a number of opportunities to commercialize this technology and leverage its broad effects. Many are listed here. The ones that are bolded, so stroke, Lou Gehrig's disease, ALS, MS, osteoarthritis, heart attack, and CLI. We have active preclinical evaluation programs in each of those areas. However, for a number of reasons, we have chosen to focus on a broader area that we will call ischemic brain injury. And the opportunity really is driven by two potential indications that we see. We've heard a little bit about stroke already, 1.3 million cases each year in the US and Europe. It is the number one cause of long-term morbidity and number 4 cause of mortality annually in the US and in the world. And the reason it is, and as I think many of you know, is that the only approved product, TPA, or Activase, can only be used in about 5% of cases because it must be administered within the first 3 to 4.5 hours after the stroke. After that, it's not effective, and it actually worsens the stroke by causing large hemorrhage. We believe this is a $6.5 billion opportunity, and our assumptions there are half the patients get to the hospital in time to receive our product. We believe 36 hours, and a $5,000 per treatment use, cost, which is a 20% cost increase over TPA. Now, there's another area that we are rapidly exploring. I'm going to talk a bit more about this later. It's a surgical procedure called transcatheter aortic valve replacement, or TAVR. It's an alternative to open heart surgery for patients with aortic stenosis, basically patients who are too frail for the open chest procedure. About 59,000 cases in the US and Europe, growing very rapidly in the US, about a 22% CAGR, a little slower in Europe as it was adopted earlier there. The challenge, it's a wonderful procedure, and if you've known patients who've had this, it has a dramatic effect on quality of life. The drawback, though, is that 60 to 80% of patients who have a TAVR procedure will suffer a silent ischemic stroke. Won't be clinically evident. About 1 to 5% of patients will have a clinical stroke. But up 2/3 to 3/4 of patients will have a silent stroke that's measurable by MRI. And those are important because an accumulation of those events over time will certainly lead to cognitive decline. And you can also tell the importance of this. About 25% of the ongoing trials on clinicaltrials.gov where TAVR is the procedure, a quarter of those procedures those clinical trials are actually focused on how we reduce this morbidity effect of throwing off some plaque with baskets or screens. We believe this is just about a $300 million market opportunity annually in the US. So why stroke? Why did we start off with this ischemic brain injury? Well, the science really led us here. So this is a model and some 3 slides from a neonate mouse stroke model. Normal architecture on the left-hand graphic. In the middle, the animals that where a stroke was induced. You can see the massive reduction in the size of the neuronal area there in the middle, or on the left-hand side. And then the panel on the right, animals where our product, NFX 101, was used, really a significant protection of the brain there. So a couple more data slides here. The slide on the left is from that same study. We're showing a dramatic reduction in the loss of neurons in these animals. What's not shown here, but is important to note, is that we had about a 50% improvement or protection in terms of functional capability of these mice as well, with treatment up to 36 hours. So importantly, the TPA window, 3 to 4.5 hours, 24, 36-hour administration of our product that was shown to be effective. At 48 hours, it wasn't effective. So it's important to know, I think, really from showing a dose response that if you treat too late, we see less effect. Importantly, on the panel on the right, a very strong dose response. This is an adult mouse model, very strong dose response. So again, giving credence to our belief this is truly a drug effect and not just some natural response in the treated animals. So our business plan, the way we plan to create value out of in the ischemic brain injury area is right now we're in the midst of conducting our IND enabling studies. We'll meet with the FDA early next year for a pre-IND meeting. But our intention is to submit our IND for right around the middle part of next year, end of second quarter. And then we'll head into a phase 1/2 clinical trial in TAVR patients, about 40 patients we anticipate. And what's really, we believe is really important about the study is not only will it give us the ability to show safety, but also it has the opportunity to provide an important efficacy signal as well. Assuming positive results there, then we'll head into a phase 2 trial in the TAVR procedure. And then once that's complete, then we'll further investigate the stroke indication and go for market approval for the TAVR procedure. Importantly, from an investor standpoint, we believe as soon as we're able to show an efficacy signal in a TAVR situation that we will be able to start to talk potential exits or partnering events, and that certainly will increase in opportunity as we complete the phase 2 clinical trial. Now, I mentioned earlier on this is a platform technology in our view. And we've done work in a number of different areas. I'm showing really quickly a couple of slides here of work that's been published within the last month. One was the panel on the left is showing a doubling of survival time in mice with ALS. And on the right-hand panel, rabbits in the critical limb ischemia model showing the green line, an improvement in perfusion of the hind limb in these animals back to essentially baseline levels. So really interesting data there. And the way we're going to leverage this is that over the course of the next year, while we're conducting our phase 1/2 trial in TAVR, we will be conducting a number of preclinical evaluations in a number of different settings with the goal then of selecting a lead or a secondary indication at that point in time. This work will all be funded through grants. And then we'll file an IND for selected indications in late 2017 will be our plan as time we have safety data from our phase 1 trial and we're able to raise additional money to pursue additional indications. Phase 1/2 trials then will follow that. And those phase early clinical trials will be funded, we believe, by our Series C financing, which will be coming up in about a year and a half from now. So let me just summarize where we are. So from an investment standpoint, we are currently raising a $3.5 million Series A investment, about $1.45 million, not about, $1.45 million is available for new investors. So we've had great success in raising money until now. This is a preferred stock offering at a $4.7 million pre-money valuation. What I probably should have said first in this slide is the key milestone that we'll achieve. This will enable us to initiate our clinical trials following the IND approval that we anticipate this summer. So our team, real quickly, we'd like to introduce them. So my name, again, is Wade Lange. I've spent my career in the pharmaceutical industry, starting at Lilly, and then management positions in the contract research business at West Pharmaceutical Services in the last 7 or 8 years, running a biotech company in Indianapolis called ImmuneWorks. Many of you know probably Keith March, my colleague and founder of the company. Keith is a professor in cardiology and biomedical engineering at Indiana University. The discoverer and a long history of inventorship, productive inventorship in this world. Importantly, a product by the name of Perclose, which is eventually purchased by Abbott. Brian Johnstone is our lead internal scientist, a cell biologist, formerly at Sangamo Biosensors. Jennifer Rhodes is our chief business officer, and Karen Momper heads up our business development operation. Karen coming out of Bristol Myers Squibb early in her career as well. So really a great team with great experience that we're going to be able to take this asset forward and bring great value out of it. So we're in a meeting all talking about regenerative medicine, how it's game-changing. We believe we're bringing to the table another step in the advancement of stem cell therapeutics that will truly help change the game in therapeutics and in our healthcare. Thank you very much, and if there are any questions, please answer them now or after the meeting. Thank you.